All living organisms are composed of numerous coordinated small compartments called cells. A cell may be defined as the smallest unit of structure and functions of living organisms. New cells are always produced by the division of pre-existing cells. These new cells need to be produced for growth, development, replacement, repair, and reproduction. It means cell division can be defined as a method of producing new cells from pre-existing cells by their division in such a way that the daughter cells receive the same hereditary materials, that is, chromosomes. That is also known as a DNA as present in the parent cell. Is of two types, mitosis and meiosis. Mitosis. The cell division leading to growth development, repair and replacement is called mitosis. Meiosis. The cell division leading to production of gametes or sex cells is called meiosis. Every cell capable of division passes through cell cycle which consists of two phases. Interphase, that is resting or non-dividing phase. It is also known as first phase. Mitotic phase or mitosis, that is dividing phase or also known as M phase. Mitotic phase or mitosis, that is dividing phase, also known as M phase. Mitotic phase is also known as mitosis. It is the dividing phase during which nucleus as well as cytoplasm divide into two daughter cells. It is a type of cell division during which a cell divides into two daughter cells containing similar and same number of chromosomes as present in the parent cell. Hence, it is also called as equational cell division or somatic cell division. It is studied in two parts. Karyokinesis, that is division of nucleus, and cytokinesis, that is division of cytoplasm. During karyokinesis, many events takes place in a sequence which can be studied in 4 stages. First is prophase, second is metaphase, third is anaphase, and fourth is telophase. Prophase. Pro means first. It is usually the longest and most complex phase of mitotic cell division. It lasts for about 50 minutes. It begins with the shortening and thickening of chromosomes. Each chromosome splits longitudinally to form 2 chromatids, which joined at one point called centromere or kinetochore. The centriole in the centrosome divides into 2. The animal cells may contain 1 or 2 centrioles in the centrosome. The dividing animal cell contains 1 centriole, which splits into 2 in the early prophase. Centrioles develop very fine eyelash-like threads called astral rays. Centrioles along with astral rays are called asters. The 2 asters start moving towards the opposite poles. By the end of prophase, they reach at the opposite poles. Between the 2 asters, very fine proteinaceous structures called spindle fibers are developed. Such a spindle is called amphiastral spindle. In plants, the spindle is without asters and is called anastral spindle. The chromosomes get attached with the spindle fibers from their centromeres. The nuclear membrane and nucleus starts disappearing in the middle of prophase and totally disappears by the end. Metaphase. Meta means after. Metaphase is short and simple. It lasts for 2 to 10 minutes. The chromosomes arrange themselves between the poles on the equator of the spindle. The chromosomes so arranged form a plate-like structure called equatorial plate or metaphase plate.
Anaphase. Each centromere splits into two daughter centromeres, each retaining one of the two chromatids. The structures so formed are now known as sister chromatids or daughter chromosomes. Sister chromatids, that is, daughter chromosomes of each pair so formed, start moving towards the opposite poles. This movement is perhaps brought about by the contraction of spindle fibers. By the end of anaphase, out of each pair of sister chromatids, that is, daughter chromosomes, one reaches at one pole and the second at the other pole of the spindle. Telophase. Telo means end. Each sister chromatid, that is, daughter chromosome, which reaches at the opposite poles, starts uncoiling, thinning, and elongating. They form a network-like structure called chromatin reticulum. Around each chromatin reticulum at the two poles reappears nuclear membrane, giving rise to the two daughter nuclei. Nucleoli reappear in each daughter nucleus. Spindle fibers, astral rays disappear. The centrioles duplicate. They are surrounded by centrosphere, again by producing centrosome near each daughter nucleus. The process of karyokinesis is followed by cytokinesis.
Cytokinesis, that is, division of cytoplasm. Division of cytoplasm takes place due to which each haploid cell completely divides into two haploid daughter cells. Hence, in all, 4 daughter cells are produced. Each daughter cell has half the number of chromosomes as compared to the parent cell. Significance of meiosis. Reproduction. Maintenance of chromosome number constant. Variations. Then crossing over. This is a phenomena in which the maternal and paternal chromatids of the homologous chromosomes during first reduction division link together to form chiasma, through which exchange of genetic material takes place, resulting in genetic recombination. That is, it produces new combinations of genes. This combination provides innumerable variations in the progeny due to which children of same parents, however similar, are different from each other in certain respects. Meiosis. It is the kind of cell division that produces sex cells or gametes. It takes place in the reproductive organs, that is, testes to produce sperms and ovary to produce egg or ovum in humans. In plants, it takes place in anthers to produce pollen grains and ovules in ovary to produce egg or oosphere. The number of chromosomes in this type of cell division is reduced to half in the daughter cells. Hence, it is called reductional cell division. Meiosis consists of 2 separate divisions of sex cells. Meiosis 1, that is heterotypic or reductional division. This division is meiotic, in which the chromosome number is reduced to half, that is N. Meiosis 2, that is homotypic or equational division. This division is mitotic. Each of the 2 cells formed in meiosis 1 further divides into 2, resulting in the formation of 4 daughter cells, each with haploid, that is N number of chromosomes. Meiosis always takes place in diploid cells. A diploid cell is one in the nucleus of which the chromosomes are present in the form of pairs. That is, each type of chromosomes are 2 in number. One is maternal and the other is paternal. Difference between mitosis in animal and plant cell. Animal cell, plant cell. spindle formed is amphiestral, whereas in plant cell, spindle formed is anestral. Cytokinesis by constriction, that is furrow method, takes place in animal cell, whereas in case of plant cell, cytokinesis by formation of cell plate takes place. Cytokinesis proceeds from outside to center in case of animal cell, but in case of plant cell, cytokinesis proceeds from center towards periphery. In animal cell, centrosome is present, so two esters are formed. But in case of plant cell, centrosome is absent, so no esters are formed. Difference between mitosis and meiosis. Mitosis occurs in somatic cells of haploid as well as diploid organisms, whereas meiosis occurs in reproductive cells of diploid organisms only. It produces 2 daughter cells. Meiosis produces 4 daughter cells. Mitosis is completed in one division, whereas meiosis is completed in 2 successive divisions. Mitosis is equational division, whereas meiosis is reductional division. Genetic constitution of daughter cells remains the same in mitosis, whereas it becomes different in meiosis. No synapses takes place in mitosis, whereas synapses does occur during prophase 1.
No crossing over takes place. Crossing over takes place in prophase 1.
Mitosis takes place during growth, repair, regeneration and development of an organism, whereas it takes place during reproduction of an organism in case of meiosis. The carriers of heredity. Chrome means colored and soma means body. These are small but thick, rod-like, darkly stainable structures, seen in the nuclei of dividing cells only at metaphase and anaphase stage of cell division structures, called chromatin reticulum. Chromosomes were first of all discovered by Hofmeister in the nuclei of polar mother cells of Tradescantia in 1848. He called them nuclear filaments. These nuclear filaments were called chromosomes by Waldeyer in 1888. Chromosome number. The number of chromosomes always remain constant in the cells of each species. Depending upon the number of chromosomes, the cells are said to be haploid N or diploid, that is 2N. The 2 chromosomes of one pair are exactly similar in shape, size, structure and bear genes controlling same characters on the same loci. Such chromosomes are called homologous chromosomes. Out of each pair of homologous chromosomes, one is shared by the female parent, that is maternal chromosome, and the other is shared by the male parent, that is paternal chromosome. Diploid number of chromosomes in some species. Name of the species: 2N number of chromosomes. Ascaris, 2 is equal to 1 pair. Drosophila, 8 is equal to 4 pairs. Frog has 26 number of chromosomes, that is 13 pairs. Lion, tiger, domestic cat has 38, that is equal to 19 pairs. Rat has 42, that is equal to 21 pairs. Rabbit 44, which is equal to 22 pairs. Man 46, which is equal to 23 pairs. Gorilla has 48, that is equal to 24 pairs. Fowl dog has 78, that is equal to 39 pairs. Mouse has 40, that is equal to 20 pairs. Maize has 20, which is equal to 10 pairs. Onion, 16, which is equal to 8 pairs. Garden pea, 14, which is equal to 7 pairs. Crayfish, 200, which is equal to 100 pairs. Ophioblossom, which is a fern plant, 1262, which is equal to 631 pairs. The correctness and complexity of an individual rather depend upon the types of genes present on the chromosomes, not upon the number of chromosomes. Structure of chromosome. Chromosomes are highly condensed chromatin fibers formed at the prophase stage of cell division. The structure of chromosome is best studied either during metaphase or anaphase of cell division. During metaphase, each chromosome consists of two exactly similar rods called chromatids. The two chromatids are joined together at a point called centromere or primary constriction. Parts of chromatids on the two sides of centromere are called arms. In each chromatid is present a highly coiled thread of DNA and proteins called chromonia. The chromonia is surrounded by matrix, which is covered by a covering called pellicle. Besides primary constriction, secondary constriction can also be observed in some chromosomes. Such a secondary constriction, if present, gives rise to nucleoli during interphase and functions as nuclear organizer. The chromosome having nuclear organizer also possess a small fragment or knob-like structure called satellite. Such chromosomes are known as SAT chromosomes. The terminal regions on either side of the chromosomes are known as telomeres. The chromosomes consist of about 40% DNA and about 60% histones, which is a particular type of protein. The histones and DNA form a kind of complex in which the DNA strands wind around histone molecules. Each such complex is called nucleosomes. A single human chromosome may have about 1 million nucleosomes. Genes. Genes are the specific part of a chromosome composed of DNA. They determine the hereditary characters. They are responsible for the transmission of characters from one generation to another. They occupy a fixed position in the chromosomes. They are arranged in a single linear order in a chromosome. Scientists have identified around 30,000 genes in human chromosomes. Functions of chromosomes. Chromosomes form the only link between the two generations. They control the synthesis of structural proteins which help in cell divisions, cell repair, and cell growth. Sex chromosomes, that is XX and XY, determine the sex of individuals. Chromosomes undergo mutations, that is sudden changes, and contribute towards evolution of life. They control cell metabolism. Types of chromosomes. The position of centromere varies in different chromosomes. Based upon the position of centromere, the chromosomes are categorized into 4 types. Metacentric. The centromere is present in the center and the two arms of the chromatid are equal. During anaphase, such chromosomes appear V-shaped. Submetacentric. Centromere is slightly away from the center, so that one arm is slightly larger than the other. Such chromosomes are L-shaped during anaphase. Acrocentric. The centromere is present near one end of the chromatid so that one arm is very large than the other. Such chromosomes look J-shaped during anaphase. Telocentric. The centromere is present at one end of the chromosome so that it looks I or rod-shaped during anaphase.